I'd like to transition into IVT and talking a little bit more about where you see some of the innovations in this space going or needing to be made. I think we constantly are referring to the mRNA manufacturing process as something that is much more efficient than a cell-based process, right? From a space, a CapEx standpoint, it's a lot smaller footprint. But I'd like to believe that the IVT process hasn't necessarily been 100% solved for us yet, right? I like to believe that it's like a coy mistress. It's got a few little secrets that we still have yet to learn, right? So I've heard the argument, right, that the IVT process is generally well established and less concerning than some of the downstream aspects of development. But I'm curious, to hear your perspectives on whether you believe that's true. Is it actually 100% solved for? And also, in what ways do you think it's pretty well standardized today? So let's start with John, and then we'll go on to Hari.
Okay. Yeah. So I would partially agree. I've come previously from working on vaccines, more conventional vaccines and biologics, and when you're working with biological systems, there's a level of complexity, right? You have the biology and the chemistry that are acting together, sometimes in strange ways. But I would say the basic operations, Hari kind of described it, but IVT, you have an IVT reaction, a digestion, DNase digestion, and purification steps. So I think the basic framework is pretty standard. I think there's been very good work that's been done, internally as well as externally on the kinetics of the reaction chemistry. It could be understood, it could be modeled, and there's, I say, enough of an array of analytical technologies that are out there that enable us to understand the chemistry and the kinetics of the reaction, right? And then I think where the opportunity sits for us is, how do you continue to optimize, one, having the understanding of the kinetics, and then how do you optimize your reaction? And I'm a big believer coming, from the world of biologics, that I always look to focus on the optimization of my upstream process so that I can lower the burden for my subsequent purification steps, right? So that's always the philosophy that I look to, right? And I think that's really the opportunity for further understanding and optimization.
Okay, that's a good point. I think I was just at a gene therapy conference specifically, and that was one of the big takeaways, was really get your upstream in order, right? For the downstream folks, really focus in on that optimization process. So that's a big piece.
I love my upstream and downstream team equally, but
Yes. But one can make it easier for the other, right?
That's true.
One's the older sibling that is paving the way for the younger sibling, maybe?
There's a codependency here, yes.
Hari, how about you? Where do you see things being relatively standardized? And are there areas that you think really demand a bit more attention from the IVT process in terms of optimization?
Yeah, the beauty of mRNA is that it doesn't require a cell to make to be produced. So the upstream process that we were just talking about, that John was just talking about, is significantly simpler relative to biologics, to a point where you could even make it a deterministic system in the sense that you could model it using classic differential equations, and now with ML, you could do even better. And so you can imagine that, it's a high, I am a chemical engineer, and I grew up studying petroleum refining, for example, where the sophistication of modeling is so high that you're squeezing, fractions of cents out of different process steps. And I can imagine a similar future for mRNA IVT, because it is ultimately somewhat of a deterministic process. We are not, I'd say we, the broad we, I think it is standardized at what I would call a rudimentary level. I think there's a lot more sophistication to be had across the industry And in 10 years from now, we will have applied all of the engineering principles that are very standard in other industries in IVT, which would make it an incredibly efficient and low-cost process. So I think, at a very kind of basic level, I think it's somewhat standardized, but I think there's a lot more to be done over the next several years.
Yeah. I like this notion of relying on the modeling as one of those key pieces, right? Machine learning to help us to get to the root, right? And better flesh out what our processes could look like and be more in terms of being more optimized. I know I'd really love to figure out from both of you if there are any sort of low-hanging fruit, non-secret sauce providing optimizations in terms of, when we're thinking about the optimizations that can potentially be made today for the industry. And I'm thinking sequence independent, right? There are going to be some sequence dependent as well. But from an independent standpoint, are there any optimizations that you think are really essential for upstream processing teams to be regularly evaluating today, right? In terms of their upstream processes to improve drug substance production? And again, you don't have to give us a secret sauce, though I'm sure some people would really love that. Can you answer it without giving away the secret sauce?
From my perspective, I think Hari answered the question in a sense, right? I mean, focus on the kinetic models, focus on the reaction, getting on that level of understanding, right? It's clear there, there's some inhibitory components. There's some, looking at your stoichiometry, your ratios of additions, et cetera. All these things are there. And I would say also consider in situ measurements that can give you a real-time feedback. So you can really understand not just, the end product quality, but I think it's really understanding the reaction and the influencing factors that are occurring, right?